sidewinder. Like
Yeah,
it wouldn't
yeah.
just be race with you.
Well,
Maybe a normal individual, but with him, like
take
it
little
needs.
pockets of time and you can buy those.
Yeah.
They do, but I don't know if you're eligible for it. How would that work?
So it's been so much strange. I'm just like,
okay, we're going to go ahead and get started. It's really a terrific pleasure for me to introduce Tara Doucet-O'Hare for her thesis seminar today. Let's see now. Tara interviewed with me. She reminded me in the fall of 2010 when she was just entering grad school. She came and talked to me about doing a possible rotation. However, she decided after doing 3 rotations, obviously she didn't do the rotation with me, but she hadn't really settled on a graduate lab to work in, and so she said, well, you know, it wasn't so bad talking to him. Maybe I could get along with him and work with him, and so that's what happened. And she also decided that she needed a little female companionship, so she added Kathy Burns as a co-mentor, right?
She wasn't afraid you'd retire.
Yeah, all right.
I didn't say any of this.
Yeah. So in any case, I have to say that I'm really very proud of Tara's accomplishments. She has she's done very well. She has a first authored paper in the Proceedings of the National Academy of Science, and she's written a methods paper, and she has data for another primary first authored paper that she's going to submit in another month or so. So that's really very good. She works very hard. She's somewhat tolerant of me coming in and bugging her almost on a daily basis as to whether she has any new data. She says if she doesn't, she just tells me to go away and get on with her work. But let me see, what else can I tell you about her? She really is very proud of her Louisiana heritage, and she likes Cajun food very much. She's just gotten married about 2 weeks ago, and she's really lined up a great postdoc with Abby Nath down at NIH. So, Tara, why don't you tell them what you've been up to?
Wow, there were a lot fewer of you when I sat down. Thank you, Haig, for that generous introduction. Today I'm going to tell you guys about what I've been working on in the last 4 or 5 years with Haig in the lab, as well as with Kathy Burns. They've both been awesome advisors, and I'm very lucky to have them both. So I'm going to tell you about somatic variation and genomic instability generated by retrotransposons. So I'm going to give you a little bit of a roadmap of where we're going to go today. We're going to start out with what are retrotransposons, how do they work, and why do we study them? And then we're going to talk about a project that I did that was published that Haig just mentioned on Barrett's esophagus and esophageal adenocarcinoma. Then I'm going to tell you some more recent work that I've done in esophageal squamous cell carcinoma, looking at retrotransposition in those samples. And then I'm going to wrap it up with a little bit of hand waving and some thoughts about what it all means and where do we go from here. So what are retrotransposons? Well, there are sequences in your DNA that are able to move from one location to another using an RNA intermediate, and we consider this to be a copy and paste mechanism. They make up about 42% of the human genome. And there are 2 main types of retrotransposons. One of the types is called LTR retrotransposons. This stands for long terminal repeat. And the other type is called non-LTR retrotransposons, which of course are lacking those long terminal repeats. We primarily focus on non-LTR retrotransposons in the Kazazian lab. And I primarily work with line 1, which is the retrotransposon which is responsible for the movement of all other retrotransposons in the human genome, because it encodes proteins that enable this element to mobilize. There are also non-autonomous elements, in other words, elements that use the proteins encoded by line 1 to mobilize, and these are signs, which an example of which would be an ALU element, and SVA elements. Since I primarily focus on L1 elements, that's what I'm going to tell you a little bit more about. So they make up about 17% of the human genome. There are about 500,000 copies. However, most of these copies are truncated and therefore they're immobile because they're too short to encode the proteins that they need to mobilize. You have about 80 to 100 that are active in your genome, and they can affect genetic architecture by inserting into splice sites, transcription factor binding sites, exons, and also causing unequal homologous recombination. Because these new insertions could be detrimental to the host tightly regulates them in most cells, especially during development. It does this in 3 different ways, with methylation, RNA interference, and post-translational regulation mechanisms as well. So each individual has a different set or suite of potentially active L1 elements in their genome, and we refer to these as inherited polymorphic insertions. The distribution of these inherited polymorphic L1 insertions varies among populations, and it mirrors population substructure, much like single nucleotide polymorphisms. So now I'm going to tell you what actually the meat and the potatoes of the L1 element is. So it has a 5' untranslated region and a 3' untranslated region. It's followed by a poly A tail, and there are 2 open reading frames that encode for the 2 proteins that mobilize this element. It also has a minimal promoter in the first 100 base pairs of the element, and an antisense promoter that's also present in the 5' UTR. It's flanked by target site duplications, which are a hallmark of the process by which these elements mobilize. Essentially, it's the same sequence on either side of the element. Open reading frame 1 encodes a protein that has a coiled coil domain, an RNA recognition motif, and a carboxy terminal domain. This protein is known to have RNA chaperoning activities and binds the same RNA from which it is translated. Open reading frame 2 encodes an endonuclease and reverse transcriptase domains, and these are responsible for cleaving the DNA open in the nucleus and then reverse transcribing the RNA molecule back into the DNA, thereby completing the cycle of the insertion. So primarily we study somatic insertions in the lab, and these are defined as insertions that are not inherited from a parent. They're present in only a subset of cells. So for example, with the work that I've done, they would be present in the tumor and absent from the normal tissue, or even present in a subset of cells within the same tissue. They're unique to an individual, and they've been mainly observed in the hippocampus of the brain, as well as epithelial cancers. Interestingly, they occur with greater frequency in some individuals than in others, and this has been observed both in the brain as well as in cancer. So if, for instance, someone was looking at maybe 3 patients in looking in their brains, either with single cell sequencing or even with bulk sequencing, they might see one individual has maybe 100 insertions or even more, whereas another person may have just a handful, and then the third person may have none. This pattern is also observed in many of the epithelial cancers that have been looked at with next-generation sequencing. So we think that this is due to differential regulation of host factors that control this process in individuals, as well as the fact that individuals have different inherited polymorphic elements that have variable activity due to the methylation of their promoter, for example, or potentially their location in the genome. The elements, especially in epithelial cancers, tend to be very 5' truncated, so they're not inserting a full-length element that could then mobilize again, for the most part. They're inserting a very small fraction of the full 6,000 base pairs of the element, maybe less than 1,000 base pairs on average. So I'm going to show you just a very cartoonish example of how this might happen. So between your 2 homologous chromosomes, you may have an inherited polymorphic insertion on one of them, which then mobilizes to a new location, creating a somatic insertion. It could be on the same chromosome or a different chromosome. So I'm going to give you a little bit of information about how these elements actually move. So initially they are transcribed in the nucleus into mRNA. They're then exported into the cytoplasm, where the protein ORF1 and ORF2 are translated from this RNA transcript. Both of the proteins then bind the same transcript from which they were translated, and together as one particle go back into the nucleus, where the ORF2 molecule uses its endonuclease domain to nick open the DNA here. The poly-A tail of the RNA binds with the DNA, creating a hybrid, and reverse transcription then happens, allowing this RNA to be transcribed into DNA, and then we have a resolving of this process, which is still very unknown exactly what happens, but it is being actively studied in our field. And then you have a new insertion, which is the red DNA, obviously. So the reason that we're so interested in this process is not just because it's really, really interesting all on its own, but it does have a link to human health. So in 1992, the first insertion that was found in a cancer gene that seemed to really imply that the insertion could be causing the disease was found in a colorectal cancer patient in the last exon of their APC gene. This insertion was confirmed with Southern blot. About 18 years later, when next-generation sequencing had become more common and cheaper, people began to really evaluate many different types of cancer with regard to retrotransposition. So the first group to really do this in a robust way was in 2010, Scott Devine's group, and they looked at a subset of lung cancer patients, and I think they looked at about 22 patients, and at least a handful of them had somatic insertions. Some of them had many, and some of them had only a few. However, this was a really interesting phenomenon, and people were wondering, could it be contributing to carcinogenesis? So many other groups, including our own, have looked at this process in epithelial cancers since then, and we still have some lingering questions after all the work that's been done in the field. One of which is, we know that L1 is active in epithelial cancers, but it hasn't really been definitively shown that it has a role in cancer development. We wonder if it could be occurring in tissues that are precursors to cancer, and we wondered if new insertions and or retrotransposon protein expression could be used for diagnostic purposes. This is something that a postdoc in our lab, Sylvia Solyom, is working on very diligently to use these insertions for diagnostic purposes in the future. So our hypothesis was that somatic insertions occur more frequently in tissues that have increased dysregulation, such as precancerous lesions and cancer, and that insertions are going to correlate with genome instability and protein expression. So the way that we identify these insertions is using a method that was developed in our lab in 2010 by Adam Ewing. We essentially take advantage of some very special nucleotides in the 3' end of the L1 element that is active in humans. By using a PCR-based method to enrich for this element, we create a library sequence on the HiSeq 2500. We map our sequences back to the reference genome, and then we compare our results to everything that's previously been published with regard to polymorphic insertions and reference insertions, and we look for things that are different, that might be unique to these individuals. So a little bit more of a specific explanation of our technique. So the primer that we use has AC, which are the diagnostic nucleotides of L1, human-specific element, and they're at the very 3' end of the oligonucleotide, which confers the specificity needed for this assay. We do 5 cycles of asymmetric PCR using only this L1 primer. And then after those cycles, we add in 8 degenerate primers and then exponentially amplify the products. We follow this with a PCR that adds on the adapters necessary for Illumina sequencing. In this adapter, or this primer here, there's also a 3' nucleotide of a G, which is also a diagnostic nucleotide for the human-specific L1 element. So we are again selecting for only the active subfamily of L1s in humans. After that, we end up with a product that has 2 adapters, the flank next to the insertion, and the poly-A tail. We then align these to the reference genome and analyze our data using a set of algorithms that were designed by Adam and modified by myself. So I told you a little bit about what retrotransposons are, how they work, and why we're interested in them. I stated that they are in fact active in epithelial cancer genomes, but that the question still remains whether or not they're contributing to cancer development. So now I'm going to tell you about my work with Barrett's esophagus and adenocarcinoma. So when I first joined the lab, in about fall of 2011, when I started to do my work in earnest with Haig, this paper had just come out from Joan Curcio's group about how retrotransposition had been connected to chronological aging and genome instability in yeast. And I thought this was a really interesting article, but I wondered if there was a way that we could study the same sort of topic using human tissues. So I asked the senior graduate student in the lab, Adam Ewing, if he knew of any disorders where we could get serial tissue samples, and he suggested Barrett's esophagus. So I wondered how challenging it would be to get these samples, and of course, what is Barrett's esophagus? So, Barrett's esophagus is a disease in which there's a loose sphincter between the stomach and the esophagus, and gastroesophageal reflux disease occurs when the contents of the stomach wash back up into the esophagus of the patient. In some patients, this then proceeds to become Barrett's esophagus, where there's a change in the lining of the esophagus from stratified squamous epithelial cells to columnar epithelial cells that look more like the lining of the intestine. Those cells are more used to a high acid content, and it's believed that that's why the cells undergo this metaplastic process, so that they can better cope with a different environment. So the reason that we believed we could get serial samples from these patients is because the doctors who treat them take samples from them every 2 to 3 years from a variety of locations up and down the esophagus, and they're usually about 1 to 2 centimeters apart. So here I just have sort of a cartoon of what that would look like. Now what they're looking for is to see if the Barrett's esophagus, where the line is between Barrett's and normal, has moved further up the esophagus, as well as looking for whether or not it's progressing to dysplasia or even adenocarcinoma. Once the cancer has been diagnosed in these patients, there's usually very little time left for them, and the 5-year survival rate is under 20%. So it's very important to catch it early. So in this patient, for example, sample 5 up here, you may see simply normal esophagus, and then at sample 4, you might see something more borderline where there might be inflammation or some metaplasia beginning. Samples 2 and 3, you would probably see mostly the metaplastic tissue, and then sample 1 here, you might see something like low-grade dysplasia if their disease is in fact progressing. So this is what it looks like on a cellular basis. Here on the left, you can see what the normal stratified squamous epithelial lining looks like of the esophagus, and then as you progress toward invasive carcinoma, you can see that these glands are forming, and they get more and more irregular, as do the nuclei, as the cancer or the disease progresses toward cancer. So with the Barrett's esophagus, you can see these goblet cells here forming and the beginning of the glands. So that's what we would look for when looking at the pathology of these patients. So, newsflash, you can't always get what you want. It was really, really hard to get these samples. I wrote a proposal. I emailed people all over the country, asked them if they would be willing to share these samples with me. And it was difficult to get patients with the similar time intervals, and most of the time intervals are only 2 to 3 years apart. We would much rather have something maybe a decade apart so that there'd be a greater chance of seeing a new somatic event in the same individual. The other problem was there were not a whole lot of patients for whom they still had DNA. Most of the research that's done on these patients, their tissue samples are used to collect DNA, and then very shortly after that, they're used in a research experiment. It's not, they don't really catalog them for very long periods of time for the most part, or they may use up all of the sample in their first study. There was not a whole lot of DNA because these pieces of tissue from the esophagus are rather small, and you're not going to get a whole lot of DNA from a small piece of tissue. But if you try sometimes, you might find you get what you need. So luckily, we found Dr. Stephen Meltzer here at Hopkins, who was very kind and willing to share with us some really nice samples of Barrett's esophagus and cases that also proceeded on toward adenocarcinoma. So this resulted in a nice publication for us, and we were very happy that we got to work on this project with him. He was able to give us 5 patients matched Barrett's, normal, esophagus, and white blood cell DNA samples. Also 5 patients that had cancer. Their matched tumor, Barrett's, and normal DNA samples. And then we were able to get esophageal adenocarcinoma cases from the Hopkins Surgical Pathology Corps, which was also extremely generous to provide us with 10 fresh frozen samples of tumor and matched normal patient tissues. So our project had a couple of aims, and these were to compare the patients who had Barrett's esophagus and never got cancer and see if the activity of the retrotransposons in those patients was different from the patients who did go on to have cancer, because we would think that if retrotransposons were potentially playing a large role in carcinogenesis, they might be more active in those people who went on to have cancer. We also wanted to correlate the number of insertions with the age of the patient, which is something that we had seen in our colon cancer study. The older the patient was, the more insertions they tended to have. And finally, we wanted to assess the general incidence of retrotransposition in esophageal adenocarcinoma patients to determine if it's a common phenomenon in this disease. So this is a somewhat busy diagram that I'll break down for you. It's a Circos diagram of the sequencing data for one of our groups of one of our libraries. So essentially around the outside you'll see these blue rectangles. Those represent the chromosomes, number 1 through 22, X and Y. The green bars on the outside of that are representative of the reference insertions that we detected. So these are all the L1 insertions that are mapped in the reference genome, and we expect to see between 800 to 900 in any given library. Now, we only observed 752 here, but that still tells us that we observed most of the variation that was available. So we know we missed some, but we still know that we saw most of what there was to observe. We also expect to see between 200 and 300 polymorphic insertions in any given library. Here we observed 218, which also tells us that we had fairly good coverage with our library. These red marks out here on the inside of the circle represent the insertions that we confirmed into tumor alone. So these insertions were predicted to be only in tumor and were subsequently validated with both PCR and capillary sequencing. The orange dots here, there's only 3 of them, are the insertions that were seen in both Barrett's esophagus and in cancer. So they were shared by both tissues. And then the blue marks here on the very inside of the circle are insertions that we were able to validate in Barrett's esophagus alone. So I'm going to tell you about how we validate these insertions. So typically, we always have a prediction of where the insertion is in the reference genome. And we design primers that flank this site. So we expect that we will use a primer here in the 3' end of the L1, using the same diagnostic nucleotides we used for our library prep. And we will pair this primer with a primer that sits in the genome, 3' of the insertion site, or the predicted insertion site. We do a PCR with these 2 primers, which we call a filled site PCR. We only expect to see a product for this PCR in the sample in which the insertion was predicted. So for example, if the insertion was predicted to be only in tumor, we want to see a band only in tumor with our filled site PCR. We want to see nothing for the normal filled site. We also do a PCR with an empty site primer, which sits 5' of the predicted insertion site. And this PCR simply amplifies the genomic DNA between these 2 primers and is sort of a control PCR for us so that we know that our DNA is good quality and our reagents are working well. We would like to see a product for both the normal and the tumor for this PCR. Sometimes insertions are difficult to amplify, or they might be in a subset of normal cells and then were amplified in the tumor. The way that we observe this is using a nested PCR. In the nested PCR, we use product from the conventional PCR that I showed you on the previous slide, and we use new primers that are closer in to the insertion site. And we do a very similar thing to what I previously described, except we're using an L1 primer with a G, which is the same one we use for library prep in the second round of amplification. We do a PCR with these primers, and sometimes we'll see a band for normal from a nested PCR that we wouldn't have seen with a single conventional PCR. This tells us that the insertion was likely present in a handful of cells in the normal tissue, and then was subsequently amplified in the tumor. So an example of this would be this PCR here at the bottom, where I did a conventional PCR on white blood cell, normal esophagus, Barrett's esophagus, and this is the filled site PCR, so we only want to see the band in the Barrett's. And then here I have the control PCR using the empty site primers, where we want to see a band for the white blood cell DNA, the normal esophageal DNA, as well as the Barrett's esophagus DNA. So another thing that we look for when we do these experiments is clonality. How many sections of the tumor have the same insertion? So we define clonal as a group of cells that have a common ancestry. In other words, they're derived from the same cell. So here's a progenitor cell. A and B are derived from the same progenitor, but perhaps A has an insertion, and A goes on to become the cancer or the Barrett's, etc. So we wondered if a somatic insertion, either in normal or in Barrett's, could then become clonal in the resulting disease, and could this be used to potentially detect metastasis later? We also wondered if many of the insertions that we were previously detecting in cancer, or that others were previously detecting in cancer, could be occurring at a low level in normal tissue, and then simply be amplified in the resulting lesion. So this is sort of what that would look like. A single cell in a tumor has an insertion. That cell is selected for amplification and maybe goes through the bloodstream and begins a metastasis. So this would be sort of what that process would look like. So the way that we test for the clonality of insertions is we chop up the tumor into multiple pieces, and we check each one with a PCR after getting DNA from them, of course. So this slide always takes a second to come up. There we go. All right. So this is what those results would look like. So we test, of course, the normal tissue to make sure that there's no insertion present in the normal. And then in this diagram, I have up to 4 sections tested. For this insertion here, you can see that the insertion is present in 3 samples of 4 tested, and for the others, it is in all 4 sections tested. So for the most part, for 20 insertions that we tested, they were present in all sections tested, and I tested 6 sections. So it seems like a lot of the insertions were clonal in the tumor. We also have seen this phenomenon a couple of times in our samples, where, like I talked about before, with a conventional PCR, which would be like A here, you see nothing in the white blood cells, nothing in the normal, and a band in the Barrett's esophagus, where you had a predicted insertion. However, when you do a nested PCR from the conventional PCR product, you see that there is still no insertion in the white blood cell, telling us that this is not an insertion that was inherited from a parent, but there is an insertion in the normal with a nested PCR, which implies to us that this insertion was present at a low level in normal tissue. We see a very similar situation here with the cancer, where there's no insertion present in normal, no insertion in Barrett's, and there is an insertion in esophageal adenocarcinoma. However, with a nested PCR, we then see a band for normal, Barrett's, and tumor. The reason for the size discrepancy is because the poly-A tail of the insertion was shortened during subsequent replications. This is a common phenomenon that we see with these elements. So in addition to these insertions I've talked about, we observed 4 of 5 of our Barrett's patients without cancer had insertions. 20 of the insertions were in only Barrett's esophagus. 1 of those was also present in normal with nested PCR. We had 5 patients with Barrett's esophagus and adenocarcinoma. 2 of those patients had insertions, with 24 insertions in the tumor alone, 24 insertions in Barrett's alone, and 3 insertions that were shared between the Barrett's and the tumor. 1 of those was also present in normal with a nested PCR. And finally, we had our 10 patients with esophageal adenocarcinoma, 4 of whom had insertions, with 49 insertions present in the tumor alone. We do test insertions that are predicted to be in normal only. None of them validated. There were about 12, I think, that we tested between these 3 groups. In all of the insertions we've ever had predicted to be in normal and not present in tumor, we've only ever validated 1. So it's not unexpected for us to have this finding. We think these are usually false positives that are created by chimeras that form during the library prep. So in addition to looking at insertions, we also looked at ORF1P, one of the L1 proteins expressions in the tumor. On the left, you can see the hematoxylin stain slides, and then here you can see the slide stain for ORF1P in 2 different patients with adenocarcinoma. Down here at the bottom, you can really see that the ORF1 really tracks nicely with the formation of the glands that is so common in this disease. I have an even closer up picture here where you can really see the nice cytoplasmic expression in all of these glands that are forming in this tumor. The biggest surprise, I think, for this project was seeing that there was expression in the normal tissue. When I first saw this, I thought, surely it must just be background. But after showing it to Bob Anders, the pathologist that works really closely with us, and hearing him tell me, no, it looks like it's real, it was kind of exciting because we were seeing insertions in the normal at a low level, and to see expression also makes sense. So the thing that we observed with Bob was that a lot of times the expression of the protein would be absent from these stem cells here at the bottom layer of the epithelium. But as the cells would mature and move to the lining of the esophagus, they would increase their expression of ORF1P. That's likely due to the fact that these stem cells repress the expression of ORF1P with promoter methylation of these L1 elements. And then once the cells have moved on and are maturing to become these epithelial cells, the expression is allowed to happen because it's no longer as important to keep these insertions from occurring. So to make sure that we weren't just seeing something that was only in people who had disease, we looked at the normal skin and normal esophagus of 2 patients who did not have Barrett's and did not have carcinoma. And they both showed expression in the epithelial layer of their tissues as well. So we also tried to see if we could correlate the expression in these patients with their insertions. And we really didn't have any sort of positive correlation. The only observation we could really make was that ORF1 was expressed in all of our patients, although at varying levels, and that every patient that had an insertion had ORF1 expression. But there was no positive correlation there. So you may be wondering, what about insertions in genes? Well, we saw a significant enrichment of insertions into genes that were previously associated with cancer using Fisher's exact test. But this has been previously observed by other studies, so it wasn't all that surprising. This is the list of cancer genes into which we saw insertions. None of them were exonic. They were all in introns. Most of them were in really large introns, some were in the very middle, so they didn't seem to have a strong chance of actually affecting gene expression. But we have some candidates we can evaluate later if we choose to. So our conclusions for this part of our work were that we see somatic insertions in Barrett's and in tumor that's absent from normal esophagus in at least half of the people we've looked at, that patients who have Barrett's and then go on to have cancer did have more insertions on average per sample, but we're only looking at 10 people, so it's not really a statistically significant observation. It's just sort of an interesting observation. We saw both clonal and subclonal insertions that existed in Barrett's and in adenocarcinoma. We did not see a correlation between insertion occurrence and any other factor, for instance, age or disease progression or protein expression. And then ORF1P is expressed in all cases and in the squamous cells of many normal esophageal tissues. So now I've told you about what retrotransposons are, how they work, and why we're interested in them. And I've told you a lot about retrotransposition in Barrett's esophagus and adenocarcinoma. And now I'm going to tell you some more about retrotransposition in a different kind of cancer, esophageal squamous cell carcinoma. So esophageal squamous cell carcinoma has a very high incidence reported in what is called the Asian esophageal cancer belt, which is northern China to the Middle East. And there it's about 100 and 100,000 annually. In the United States, it's only 4.4 people per 100,000, so it's a significant difference. This is a comparatively rare disorder in the United States. It is the most common form of esophageal cancer worldwide. It's more common in African American men in the United States, and it typically occurs in the mid to upper esophagus. The average age of onset is between 60 and 70 years of age. The average for our patients was 68. Smoking and alcohol abuse are risk factors, and they have strongly synergistic effects. Over 90% of the cases that are observed here in the United States can be attributed to either smoking or drinking, or both. And it's normally diagnosed at a late stage with a very low overall 5-year survival rate. So, like I said, this cancer is comparatively rare in the US, so we were very fortunate to get 9 fresh frozen matched normal and tumor samples from the Johns Hopkins Surgical Path Core. And we wanted to sample these to characterize the incidence of retrotransposition and compare it to our matched normal samples. We also wanted to see if we could correlate the number of insertions with age or severity of disease or L1 protein expression. And finally, we wanted to determine at what frequency insertions were maybe occurring in normal tissue and then being amplified in the tumor. So we were able to validate 75 of 92 attempted somatic insertions. And this is a validation rate that's comparable to what we've seen before in our own lab as well as in other labs. 8 of the 9 patients looked at had insertions. 9 of the 75 events that were validated appeared in the normal tissue following a nested PCR, which is much larger than we would have anticipated. This is a 12% of the insertions that were present in normal with a nested, as opposed to 1.6% of what we observed in esophageal adenocarcinoma and Barrett's. And the total that we've seen in all cancer samples analyzed by L1-seq in our lab is 5%. Only a third of the somatic insertions that we detected in the tumors were actually amplified with a conventional PCR. So in this cancer, I would say that many of the insertions may be happening later in cancer development because they're not as easy to amplify. It could also be possible that for whatever reason the PCRs were not optimized and the insertions were just difficult to amplify. Oops. Okay. So this is an example of 2 of the insertions that were appeared in normal with a nested PCR. So you can see that in oops. These big pictures cause problems. There we go. Okay, so you can see in normal esophagus that the insertion is not present. However, it is present in the tumor, and just kidding. Everyone hold your breath. Okay. And both of our control PCRs are working nicely. With a nested PCR, you can see that there's a band present for both normal and tumor for the insertion. And this is essentially the same situation here in the PCR adjacent. Okay, so we were able to do some really nice staining, which was primarily done by Rima Sharma. So she's not here today, but big shout out to Rima. She's an amazing IHC person. So we were able to stain, and we saw really nice normal stratified squamous epithelial expression in both of these cases, as well as very clear cytoplasmic expression in the squamous cell carcinoma in the other part of the tissue. However, here we go. The most interesting thing we saw when we looked at the IHC of these samples is that there were 3 different distinct patterns of staining of WRF1. So here in this sample, you can see that it looks mostly cytoplasmic. It's a low expression, but it's definitely there. And then here you can see there's much more of a perinuclear pattern where there's an accentuation of expression around the nucleus. You can really see it nicely on this in this image as well. And not only did we see multiple patterns, but we saw them in the same case. So here in the yellow circle, you can see a large group of perinuclear staining cells here, and then in the red circle, it's mostly cytoplasmic staining. And this is, right adjacent to each other in the same cancer case in the same piece of tissue. Okay, so this is an example of really this sort of globular, if that's a word, staining around the nucleus in some of these cases. And we would really like to look more at this staining pattern to see what the protein may be associating with, if it's piling up in an organelle, or if it's just maybe being processed by the proteasome or something like that. So we're going to do some co-localization experiments to look at this more. But we've never seen this in cancer tissue before, and this cancer type is the only one that we've seen it in. And this antibody has been used to do IHC on at least 20-something cancer types. So it's very interesting. And again, we looked at the normal tissue of the stratified squamous epithelium and saw a very similar pattern to what we'd seen before, where there was very little to no expression of WRF1 in the stem cells, and then increasing expression as the cells matured and became the lining of the esophagus. So as far as insertions into genes into these samples, we had 33 of the 75 insertions validated into genes. There were insertions into genes that were previously associated with cancer. We did have one insertion very near to an exon of this gene. And interestingly, this gene has been connected to the immune response in the throat, oral cavity, and upper digestive tract. As for future directions for this project, we would like to stain these squamous cell carcinoma slides with a second ORF1 antibody from Japan to see if we can recapitulate the pattern and really confirm that this perinuclear staining is not just an artifact and is in fact consistent. Oops. And we also want to use some different antibodies for co-localization experiments to determine what organelles might be harboring ORF1 in the cancer cells that have these aggregates of the protein. So I've told you about what L1s are, how they work, and why we're interested in them. I've told you about Barrett's esophagus and adenocarcinoma, and now I've told you about squamous cell carcinoma and retrotransposition, and now I'm going to do some hand waving. So we have some lingering questions. We still want to know what role these insertions are playing in tumorigenesis. It's still somewhat unclear, even after all the work that we've done. And we want to know if cancer promotes retrotransposition or if retrotransposition might promote cancer, or if both could be possibly true. We really want to know what the three staining patterns of ORF1P mean in esophageal squamous cell carcinoma, and in which organelles the protein could possibly be accruing. We also would like to characterize how active retrotransposons are in normal epithelial tissues. Since we're seeing it at a low level in normal samples, we would like to know if cancer is really only amplifying somatic events that are already present. And we would like to know also if insertions happening in somatic tissues or early cancer development could be used to detect cancer recurrence or potentially look at metastasis in patients. So Haig asked me to talk a little bit more about what I really think the answers to these questions are, so I'm going to. So I think that the variation between individuals is likely the key in this situation because, as I said before, different individuals have different polymorphic elements that all have different levels of activity. They could have different levels of methylation as well, which regulates the activity of these elements, as well as host factors that regulate L1 activity. I think the dysregulation in cancer may be promoting L1 activity for sure, but I also think that the activity of specific inherited polymorphic L1s and the resulting insertions likely contributes to cancer development in some people, probably not in everyone. I think we really need larger groups of samples to really characterize this well. And in one paper, it was said that L1 insertions are responsible for Mendelian disease in approximately 0.27% of patients on average. I think as far as cancer goes, it's probably at least the same amount that retrotransposon insertions are causing cases of cancer, maybe greater. And now for my acknowledgments, I would sincerely like to thank Haig and Kathy for being amazing mentors, extremely supportive, no matter how panicky I was. I'd really like to thank Dave and Sandy for all of their support over the years. They've given me a lot of career advice as well as life advice that's really been very helpful. I'd like to thank Steve Meltzer for his donation of the amazing samples that he was able to give us, as well as Norman Barker for helping me take all of the beautiful pictures of IHC. I'd like to thank my thesis committee members, Bob, Jeff, Sarah, and Dan. Thank you so much for all of your help. And of course, the PhD program. It's been really amazing, and I know that I picked the right program. I couldn't have chosen a better one, and it's just been an incredible part of my life that I would be very sad to leave. But I feel like you've prepared me really well, so I think that's good. I'd like to thank my lab for all of their support and constructive criticism over the years. They've been challenging, but they've always been very helpful. And I'd also like to thank the Burns lab, especially Lindsay and Rima. Rima was the person who did a lot of the staining with me and was just really very proficient and very willing to help me. And Lindsay helped me write my first paper. She read it maybe 8 times. I don't know. I feel like I should buy her a car.
I wouldn't complain.
But she's been a great role model for me as a scientist and an even better friend. So thank you to everybody that's helped me, and I really appreciate all of your help. Thanks.
Questions?
diagnostic lab experience, I found patients where they had insertions like directly in the gene and disrupted it, and that's how they contributed. Do you, like, is there in that paper or in your experience, do you think that L1 will contribute in a way that is not just screwing up like existing genes?
There's several mechanisms by which they can cause problems. They've been incorporated into exons. They cause a lot of non-allelic homologous recombination in the genome. I think that they can also, there's also some evidence that because of the methylation in their promoter, that when they insert into a new location, the methylation can kind of spread and suppress transcription that way. So I think there's probably many different ways that they can.
Are there examples of it, like inserting into like a tumor suppressor or something and causing cancer that way?
The APC example is the one that stands out most in my mind. There were some germline insertions that were found by Faulkner's group from Australia in the MCC gene in hepatocellular carcinoma. There's also an insertion into P10. I forget what kind of cancer that was, but do you remember, Hayden? Head and neck. Okay. So the insertion into right, yeah. So it would have to be by a different mechanism like splicing or something like that.
So the histology you showed a lot of the ORF1P, I guess it is, in the cytosol of these cancer cells. What do you think it's doing in there? And since it has, in particular, since it has RNA binding capacity, do you think it's sort of perturbing the transcriptome of those cells?
It definitely could be. It can bind lots of different types of RNAs. So I think the reason there's so much of it is because it's not being well regulated and it just sort of accrues. And maybe the cell doesn't have the capacity to get rid of it as well as it normally would. I have no idea why it's aggregating though. That's we've seen different types of aggregations in cells of this protein before, but they've usually been smaller. Like John has observed in stress granules, and it has been observed in this sort of pattern in cultured cells as well from endogenous L1 expression, but it's definitely been observed a lot in cultured cells with exogenous expression too. So I'm kind of curious to see if maybe it will tell us something new about the relationship between retrotransposition and cancer and how these proteins might be playing a role we may not have previously thought of. Does that answer your question?
Mm-hmm.
Okay.
It seems like you could actually pull down that protein.
Yeah.
And see if you have RNA associated with it.
Well, that's been done quite a lot. There's a lot of different RNAs that bind it. I don't remember anything more specific than that offhand, but. Yes.
Do you have available tissue that's of a quality where you can sort of do staining and see those different patterns and then see different gene expression level or something of the cells that have one pattern of L1 staining versus the other?
So I haven't looked at the frozen sections of this. So the staining was done on paraffin-embedded formalin-fixed tissue. But we do have frozen sections of this tissue, and I have actually cut it and stained it with hematoxylin and eosin before, and it looked okay. It didn't look nearly as beautiful as the FFPE. Because Bob and I looked at all of the samples before we did any library prep to make sure that tumor was only tumor and normal was only normal. And we cut off necrosis and everything as well. So I think that we could probably do that, and Bob's also mentioned that we could do like laser capture or microdissection if we wanted to maybe look more closely at those cells that are harboring the aggregates, but we haven't really made a plan going forward yet to do that, but it's definitely on our radar. Yes?
Next topic. I have one question. I think I might have missed something. So when you showed the immunohistochemistry of the ORF1P, you showed that it was in both the esophagus, so you saw the Barrett's esophagus, you saw it there, but then when you looked at the normal tissue, you saw it there as well.
Yes.
Isn't that kind of a contradiction there if you think that that's kind of causative in this, that the ORF1P is present in the normal tissue? Or
not necessarily. I mean, I honestly think that retrotransposition is probably happening at a very low level in normal cells. Just because you have the protein doesn't mean the process is actually happening either. So the protein has to be expressed for the process to happen, but it's not necessarily sufficient. So it makes it more likely, but it doesn't, say that it's absolutely occurring unless we detect an event, and then we know.
Like the levels of ORF1P, like if it was lower, like if it was just like a baseline low level in the normal tissue and then it increased in Barrett's?
Yeah, it definitely is a higher expression. Well, we didn't have any Barrett's tissues that we were able to stain, but it's definitely more highly expressed in the tumor than it is in the normal, and that's been validated by pathologists that have looked at it, not just from me thinking that they look darker. So I feel fairly confident that that is true. However, IHC isn't necessarily quantitative, so there's no way to say for sure whether or not how much higher it is. Or
Okay, cool, thanks.
Okay. I think Tara wants to.
I have some great pictures of Hague.
show her. but I wanted to tell you that the reception following the pictures will be in room 125 on the first floor of this building. Room 125.
Okay, so I emailed Hage's wife so that she could send me some really nice pictures of Hague. I have to tell you though, my favorite one is this one right here. It's really nice, wearing some shapely pants there. She called this email a very productive meeting, which I thought was hilarious because all the pictures she sent me are Hague skiing. Also, this is a nice picture from Bar Harbor. After our first year of grad school, Hague took us on a, quote, leisurely hike that was supposed to last a half hour. 4 hours later, we're climbing down a somewhat sheer cliff face with him, quite terrified. This is a picture of the first time Hague sang karaoke in Taiwan. Here's his wife singing with him, and I don't know who these two gentlemen are, but they seem to be having a good time.
I think we're singing You Are My Sunshine.
Yes, she did say that. I don't know who this guy is with Hague, but Hague seems very focused. Oh, okay. Okay. And then this is a picture of Hague and I at Bandelier when we went to the Keystone Conference last year in Santa Fe. We climbed some very tall ladders. It was a little bit of a nail-biter there for a minute. But it was a really good time, and I really enjoyed all my hiking with Hague and all the stories he's told me. And I'm very fortunate to have worked with him. So I also want to thank, of course, the Burns Lab. So these are some pictures from when we went to a meeting in West Palm Beach this summer. We did draw the L1 life cycle in the sand using shells and seaweed, but I don't have a picture of that, unfortunately. Dan does, and he's on his honeymoon. So, and then here's some pictures of Juan, and this is Nemanja, who worked with me on the Barrett's paper. He is a pathologist. Fully trained pathologist, I think, now at Yale, right? And he's a great guy too. And then this is the whole Burns lab down here, and they've really just been very awesome. They let me come to all their lab meetings and eat all their bagels, and they help me with all kinds of things when I run out of supplies or just need a pick-me-up. So thanks, guys. Really appreciate it. These are some pictures of my classmates. This is my favorite. I found this gym on my computer and almost fell out of my chair laughing. If you want to see how this works, I don't know if Corey's here, but I know Nicole is, so she can always show you later. I'm not that good at it, but it's like a clapping thing. Anyway. Corey taught us. I don't know. This is from Bar Harbor when I made jambalaya for everybody. Like Hague said, I love my Cajun heritage and I like to share food as much as possible. Unfortunately, I put way too much cayenne pepper in that time, and I had a hard time eating it. This is a nice picture from the Jackson Labs with our class and Dave. And then this is from a recent reception I had at my house when I got married to my husband, Steve. And it was really awesome to have some of my friends come out. And also we had a really good time rafting. We've done that a couple times in Bar Harbor and then again. So I really appreciate my class. You guys have been super supportive, and I feel really lucky to have had all of you come in at the same time as me, and I've learned a lot from you. So thank you. So I would like to thank all my friends, my wonderful support system of friends that are absolutely awesome. Becca, Lindsay, Sam, Kristen, my friends Emily and Tommy, and my friend Amelia as well. Everybody's been really supportive when I called you to panic about some experiment not working, or my gel fell on the floor and broke into 10,000 pieces. You know, those kinds of things. So thank you guys for being so awesome. My amazing family. So my dad unfortunately could not be with us here today because he passed away when I was 11. But he was a really great man. He was an attorney, and he was very smart, and he always pushed me to be the best that I could be and to learn as much as I could. And I think that instilled in me a desire to learn and an excitement about learning. And so I really appreciate, the fact that he had that role in my life. And of course, my awesome mother, my brother, and my half-siblings who live all over the country, and my wonderful grandmother as well. And all my extended family have been very supportive throughout this process. They actually paid attention when I told them not to ask me when I'm graduating every 5 minutes, so that was nice too. And then of course my amazing new family, after I've gotten married, I have a wonderful mother-in-law and father-in-law that have also been supportive. I've been with my husband since I started graduate school basically, so they've known me through this entire process, and they still like me, so that's great. And then of course my brother-in-law and sister-in-law, who are also awesome people, and I'm just very lucky to have them all in my life. And I wanted to say a few extra special words about my mom, because she's my best friend and she's a wonderful parent. And I think she's probably learned a lot she didn't want to know about science, hearing me practice for this. But she may have understood more because of that. But yeah, she's been awesome though. She's always been there to talk to me when I've had a stressful day in the lab. She's always told me that she knew I could do it and just had to keep going and to be confident, and I really appreciate that. And of course my husband, Stephen, who, like I said, has been with me since the beginning. We've gone caving together. We've been to lots of people's weddings, and now we're finally going to have our own in the Dominican Republic next week. So we're very excited, and this is our child, Sophie. She's very furry. And I just really appreciate all the support that you've given me over the years. You're the best husband anybody could ever ask for, and I just love you so much. So thank you. And let's have some champ.
Very nice job.
Congratulations. This is great.
Nice job.
Yes.